It's a pleasure for me also to be here with this group this morning, and I know that some of you have heard me speak about plant pathology and the implications for biological security before, but this is a new context, and it's my job to try to speak about gene drives and gene drive technology applied to an agricultural setting. And for me, because I'm a plant pathologist, most of my comments this morning will be referring to crop-based agriculture, but also to natural plant resources like forests and rangelands that are not controlled by humans as much as the crop systems are. To begin with, I wanted, for those who haven't heard me speak before or others on crop vulnerability, I wanted to give a little bit of background to place this topic in a framework. So after the anthrax attacks of 2001, there was a lot of reflection on capabilities and vulnerabilities in the U.S., and I give a lot of credit to federal agencies for recognizing that critical infrastructures included agriculture. The National Academies released a report about that time that indicated some of the sources of crop vulnerability. One of the reasons that crops are so vulnerable to being affected by emerging or introduced pathogens and pests is just because of their value to the country. They generate about a sixth of our gross domestic product and about 17% of our employment. We know already that well over 50% of our crop losses that we have right now are due to pests or pathogens that have been introduced from other countries, and that costs us close to $140 billion a year. Vulnerability is increased by the fact that most of our plants are cultivated as crops in monocultures. So there's very low genetic diversity, and all of the plants in a given agricultural crop field have the same susceptibility to a pathogen. Another reason for the vulnerability of plants is that they occur over such huge acreages. The natural resources, the forests and rangelands that you see in these pictures, are huge, and humans go into the middle of these very rarely. But the same is true for crops that cover billions of acres. So finding something new, an unusual symptom or a new disease in the midst of that acreage is very difficult. And so as a result, there can be a long period that occur between the time that a new pathogen is introduced and the time that someone detects it and begins to do something about it. We also have pathogens that come into the country in a variety of ways every day. They come in on weather fronts like hurricanes. They come in with fresh produce that we're introducing more and more of into our diets in the United States, in the bilge water of ships, in the wooden pallets that we ship other materials on. They come in on the shoes or clothing of air travelers or even in luggage if someone wants to do harm intentionally. And then, of course, new pathogens can also emerge within our borders. Over the centuries, plant diseases have caused tremendous types and numbers of impacts on human health and sociology. Most of us are aware of the Irish potato famine in the 1880s, but when we think about that kind of impact today, certainly in developed countries like the United States, a plant disease is unlikely to cause malnutrition or even significant shortages of particular foods, but that may not be true in underdeveloped nations in the third world. And we've had evidence of the continuing impact of such shortages by food riots that have happened over the last decade or so, Arab Spring, etc. We also have concerns for human health because some plant pathogens are actually also infectious to humans, usually opportunistic or nosocomial rather than direct. But mycotoxins, which are produced by a number of plant pathogenic fungi, are also major health hazards. I think we don't even realize how dangerous the mycotoxins can be in some cases. Certainly, in agriculture, it affects livestock, the availability of feed. And then linked to this closely is the fact that contaminated agricultural produce could be carriers of human pathogens like E. coli, salmonella, and others. And we're seeing more and more of that kind of impact as well. Plant pathogens have been considered by a number of programs as for their potential to be used as bioweapons or agents of biocrime. The motives are, varied as they are with any biological agent, threat agent. But what's different about plant pathogens is that they are easily available to most people who might have intention to harm. And also they don't pose a threat to the handler as a human pathogen would, like anthrax. We know that they actually were part of the bioweapons programs of several countries, including the former USSR and our own country, the United States. And when I say that they were part of the programs, I don't mean that they were used, but they were investigated or studied for their potential and perhaps modified to increase their potential. And more recently, notes about the use of plant pathogens, particularly wheat rust and rice blast, were found in the caves of Afghanistan. This table simply shows some of the bioweapons programs and the approximate dates in which they were operational. And I've highlighted in red the agents that were particularly directed toward plants, and this includes both insects like the potato beetle and a number of pathogens like the maize rust and potato viruses, rice blast, etc. There have been some known incidents of plant-directed agroterrorism. In World War II, Germany deployed cartons of Colorado potato beetles in southern England to try to affect the potato crop. An interesting case in the late 1980s had to do with the political control by rich coffee barons, and protesters actually imported fungus-laden coffee branches, which they hung in the trees of the coffee plantations in an attempt to influence that political infrastructure, which they did. And then in the late 90s, the use of pesticides in Palestine to affect the grape production. We're looking this morning specifically at how gene drive and gene drive technologies kind of fits into this background and potential for affecting a critical infrastructure. So in context, agriculture is constantly under pressure. We're being asked to produce ever greater yields from our crops, green revolution sort of thing. But at the same time, we have to look at increasing sustainability, and particularly in the face of challenges such as climate change. And we need to address, at the same time, many issues that go along with it or that impact it, like societal, political, economic, and environmental. Genetic crop improvement can be really good by adding beneficial traits or deleting deleterious ones. But gene editing can actually increase the capability for doing that beneficial activity by offering greater gene targeting precision. This is much greater than what you can do with traditional crop breeding, for example, and also, of course, facilitating the efficient and rapid trait deployment. What might we target in crop-based agriculture? Well, a number of different targets could be considered. Bacteria and other microbes would be one, and that could include the plant pathogens themselves, but also other kinds of microbes that affect plants, like biocontrol agents that are used fairly frequently in plant pathology work, or microbes that positively affect plant growth and that are used as supplements in agriculture. Of course, insects, and a lot has been said already in these webinars about gene drives in insects, but in the agricultural sector, these could be pests that are directly impacting the crops by eating them, or insects that can be vectors of plant pathogens. I'll get back to that. Or we can look at targeting the plants themselves, and again, much has been written about the possibility of using them against weeds for weed control, but we could target the crop plants as well for beneficial effects, or looking at our natural resource plant species, the forests and rangelands. So if we look first at bacteria and other microbes, well, of course, genome editing is a natural process for many bacteria, and CRISPR-Cas systems have been found to occur naturally in close to 50% of bacteria that have been looked at, and an even higher percentage of archaea. The gene drive system here, or genome editing, serves as a heritable immune system against viruses or plasmids that bacterium has encountered previously. The use of human-directed genome editing is less well developed for bacteria, partly because of the way that they reproduce, but it is promising, and there have been success stories in bacterial typing and strain discrimination. In the plant pathology arena, one of the bacteria that has been worked with is Pectobacterium, which used to be called Erwinia atrosepticum. This bacterium causes blackleg, which is a soft rotting disease of potato that also causes a wilt. You can see a picture of it there. But in work by Verco et al., they used Cas proteins to cleave the bacterial DNA and showed that the result was bacterial growth inhibition and...
We've lost audio for Dr. Fletcher, and we're going to attempt to reestablish it. So bear with us, everyone online, for a few minutes while we reestablish her audio.
Would you like me to call in again, or?
Okay, your audio has reconnected, so please go ahead and continue. We're going to give the presentation rights back to you again.
Okay, can you see the slide then?
No, not yet.
Okay.
So you'll need to go to Quick Start again and share your screen again, Dr. Fletcher.
Okay. How's that?
Perfect.
Okay. Should I start over with this slide? Okay.
Sure.
I'm going to
That's probably a good plan.
Okay.
I heard you through blackleg.
Yes. The Pectobacterium Erwinia atrosepticum, which causes blackleg of potato, was used in this experimental procedure by Verco et al., and they showed that the Cas proteins could cleave the DNA, causing growth inhibition and abnormal cell morphology. They found that the targeting can reshape its genome and delete or significantly truncate pathogenicity islands. So this sounds pretty promising, but there are lots of questions yet to be answered that were not in this particular study. And those questions particularly are, can these changes be driven through the pathogen population, and can the same kind of technology work for other plant pathogens, other bacteria, maybe also fungi, etc. I mentioned earlier biocontrol agents. This is something that is commonly used in the management of plant diseases, and it's defined as reducing inoculum or disease-producing activity of a pathogen by an organism other than man. Often for plant diseases, our biocontrol agents are bacterial, and the best example, the most successful one, is the use of Agrobacterium radiobacter, for controlling crown gall, which is caused by a very closely related bacterium, Agrobacterium tumefaciens. In that particular case, the biocontrol is caused by niche competition and antibiosis, or chemical inhibition. But in other cases, biocontrol is accomplished by mimicry or even infection of the pathogen in the case of some bacteriophages. Now, moving on to insects, obviously insects are one of the success stories for gene drives with the mosquito work, etc. The question I pose here is whether similar approaches can be used on vectors of plant pathogens. Many bacterial diseases and virus diseases particularly are transmitted by just a variety of different insects, leafhoppers, aphids, whiteflies, squash bugs, etc., mites. But I wasn't able to find any research published that has actually focused on these sources of transmission. So I think that's a field that may be very promising. Moving on to looking at the potential of targeting the actual plants in agriculture, this is an issue because of the requirement for sexual reproduction to drive the trait through the population. And as many have pointed out, many plants reproduce asexually. The picture here is of potatoes, and we typically plant potatoes by using seed pieces, which are just old potatoes that have been cut up so that each one contains an eye and can germinate. Other asexual means of plant reproduction are by grafting or tubers or root systems that spread and then sprout new plants. Other plants are monoecious, which means the male and female organs are on the same plant. And these are generally poor candidates for gene drives because of the lack of movement through the population. But many other plants are dioecious and have the male and female flowers on different plants. And these may be more suitable in general for gene drive technology. There's also a consideration of dissemination, not so much by natural means, but also by seed distribution systems, which within agriculture are immense and global. Now, one sort of exception to sexual reproduction with monoecious plants is that cross-pollination can occur. In nature, this occurs most often when pollen is moved by wind or by insects. But it can also be done by hand, and is in some cases if, for example, bees are not available in an apple orchard or something like that. So this increases the potential. This is a list showing just some of the important monoecious versus dioecious plants. And if we think about most of our crop plants and common fruits and vegetables fall on the left list, the monoecious ones. We see there the major grains like corn and rice and wheat. But on the right are some interesting potential candidates for gene drives. You see on there some weeds, which could be targeted for weed control, and other very important food crops like soybean, along with some fruits like pistachio and persimmon, etc. So this is certainly a consideration in targeting plants. As I mentioned, they've been considered as potential targets for weed control. And unlike most crop plants, weeds are not generally controlled by genetic means, and gene drives could eliminate herbicide resistance, making eradication more feasible. If you have ever traveled in the South, you've probably seen blankets of kudzu that looks like this picture here. And there are many invasive, very invasive weed species. But looking at crop species and other desirable plants, one of the important questions is what genes you want to target. And what we've generally targeted in genetic modification of crop species are agronomic characteristics like uniformity of the crop for better packing, ease of packing, increased yield potential, a shorter time to harvest, enhanced qualities like flavor, more nutrients like super plants, more fiber. And very important is longer shelf life for saleability. Better processing characteristics, in other words, they can be made more easily into processed products. And then very important is resistance to all kinds of things, diseases, pests, drought, cold, salinity, herbicides, you name it. And this comes into play certainly in our concerns for the future and climate change. Up until recently, the older methods of genetic manipulation of plants were the only ones available. We could introduce genes into plants using biolistics, a physical means of really bombarding plant cells with particles containing DNA on the surface. Or we can actually use a bacterium like Agrobacterium tumefaciens. I mentioned it earlier. A. tumefaciens is particularly useful because we have the capacity to replace the virulence genes with beneficial genes, encoding things that we want. And then the bacterium is still able to enter the plant and deliver the desirable genes into the nucleus where they are incorporated into the plant's DNA and are expressed there. But gene drives give us a number of advantages that we didn't have with the traditional methods. Excuse me. It's much more precise, as I've already said, than conventional crop breeding or even standard genetic engineering methods. And of course, the greater the precision, the lower the risk of unpredictable or undesirable outcomes. Excuse me, I'm getting over it. Fall cold. So it's possible to change a small subset of billions of nucleotides in a plant cell, and of course this could positively impact food security and public health globally. The challenges are that targeting plant genes is inefficient and often difficult due to the low frequencies of homologous recombination. And in the developed world, at least, most of our crops are genetically identical. That may not be true in many developing nations. And then many crops are grown from seeds that are produced under highly controlled conditions. And I do want to talk about a case in which genome editing has been fairly successful. I'm going to talk about two different studies that were published by the group of Yanan Yang at Penn State University, and he chose to work on rice, which is the most widely consumed staple food in the world and the third most produced agricultural commodity. Now, rice is monoecious, so that's something to keep in mind. What Yang's group did was to create specific gRNAs or guide RNAs. These are the elements which guide the Cas9 to the desired site in the target genome for modification or control. And they designed these three gRNAs, which you see encircled here on the slide, to pair with sequences on the genes of the rice that were followed by the required PAM site, or the protospacer adjacent motif. And by doing that, they were targeting three genes that were located, three sites in a single gene that were encoding a protein kinase. The gRNAs then introduced the mutations, and the efficiency that they achieved in this work was as high as 8%. And that sounded kind of low to me, but then they made the comment that this was in the same range of the efficiencies that have been achieved with animal cells. They concluded that specific gRNAs could be designed to target over 90% of the genes in the rice plant, and they felt that the CRISPR-Cas system could be very powerful for gene targeting and precise editing. In a follow-on study in the same lab, they were addressing the limitations of CRISPR-Cas for multiplex editing, and they created a strategy by which they could produce many gRNAs from a single gene. And these were synthetic genes were processed into gRNAs that could edit multiple chromosomal targets at the same time. This worked really well for them, and they got to multiplex genome editing and fragment deletion with stable transgenic rice plants with up to 100% efficiency. And they felt that this also could be applied broadly through living organisms. Now, in both of these cases, the emphasis was on the targeting advantage and improving that part of the process. Again, there are questions about whether this would be able to go on to the drive portion or the dissemination through the population, particularly because rice is monoecious. But one tool that I wanted to mention that Yang's group put together was called the CRISPR Plant Portal. This is an analysis and bioinformatics pipeline to assist researchers in using CRISPR-Cas for genome editing in plants. And what it does is to help the user design and construct the specific gRNAs for genome editing. And they actually give database information on 8 model plants or crops that you see here that are available for anyone to use. And you can access this at the website that I show there at the bottom of the slide. So these are really the only cases that I could find where that specifically addressed plant diseases or plant pathogens. But in looking at gene editing and drives in crop applications in general, I think it's important to look at what those risks are. What is at risk, and we're talking about the crop itself, but also the larger impact on people and the environment. Certainly site-specific risks in the laboratory, as well as during field testing, which will be out in the environment, and then post-harvest. Certainly the potential for unintended or unforeseen impacts, potential for unintended release, and intentional misuse. In thinking about regulation of gene drives in crop applications, it's been pointed out that risks and benefits of genetic crop improvement, regardless of what the technology is really associated more with the particular changes that have been introduced rather than the methods by which they were introduced. But as we move ahead with things like gene drives, it may become more and more difficult to tell the difference if you didn't know how the crop was produced. And the line between what we recognize as GM and non-GM may be more difficult to distinguish. And so one question is whether the policy should be directed at the characteristics that are achieved or the technology that produced it. And of course that would depend on where the dangerous impacts could come from. So in crops, challenges could be to identify the most important factors that could be addressable by gene drive technology and determine how the regulations would impact those, or anticipating the impacts of the regulatory actions on applications in both research and practice, or in the practice of agriculture. It's going to be really important to incorporate stakeholder input, not only because their needs are important, but also because that will help to ensure acceptance by both the agricultural community and by researchers, and then addressing the complexities at all levels, local, regional, national, and global. Achieving agricultural sustainability and food security is obviously hugely complex and challenging. What we need to look at in agriculture is the levels in the field or pre-harvest, post-harvest in storage and transport, value added in product development, and commercial in practices for marketing and trade. And these kinds of needs are multidisciplinary and will require new research that will underpin those regulations. Of course, we have the potential for intentional misuse, as Ed Yu pointed out earlier, and this has always been present. We know that any technology that can be used for good can also be used for harm. And in plant pathology, some of the practices that plant pathologists use every day, all the time, might fall into a range of what people think of as being potentially dangerous. For one, the use of agrobacterium tumefaciens, and we modify that as a tool for genetic engineering. When we do field trials, plant breeders will deliberately plant an array of different cultivars that they've developed by breeding and then inoculate them in the open to test their resistance. The use of biocontrol organisms. Sometimes there are genetic changes made to make them more pathogenic to a plant, but that's a weed. It's a plant we're trying to get rid of. Or to be more pathogenic or deleterious to a competing pathogen that we're trying to control. And then, of course, the use of plant growth-promoting bacteria, which are commonly used in agriculture, particularly those that can be added as soil amendments around the roots. And we can engineer those to be more beneficial to our plants for growth. So mitigating potential negatives in crop applications could include steps like confirmation tools, using sequencing to confirm that the targeting was precise, and having reversal drives, the ability to change or stop the movement through the population at hand in case something seems to have gone wrong. Monitoring post-release to continually confirm the precision and the absence of negative impacts. And then having the robust tools for investigation in case of intentional misuse for attribution and justice. And of course, Ed, you mentioned too the education and training for both users and students and practitioners. I do believe that scientists have a responsibility to act ethically and serve as models to create this so-called culture of responsibility and expect their groups to follow this. Another responsibility, I think, is for scientists to be at the table for the development of regulations so that they can help to assure that they're reasonable and effective and whenever possible based on science, and then to comply with them. Scientific societies can play a big role in this. I happen to belong to the American Phytopathological Society, but I know that many scientific societies have various statements of ethical approaches to the science and what's expected of their members. And I think these can go a long way for us. And I'll just finish by saying that food security is in the hands of our farmers, but also our own hands as scientists. And I'll stop there.